I don't know. All right, so you see my screen?
Yes, sir.
All right. So 2025, January paper. So the first one says that your friend has just returned from a tour of South America and brought you a bag of Jumo, right? A powdered food item. She told you that Jumo's ingredients include starch, reducing sugar, and fat. But you would like to verify this by performing food tests on the powdered item, right? So complete table one by showing the following: a description of how the test is performed, including all the reagents and the suitable apparatus. What you expect to observe the food substance is present in Jumo, and what you expect to observe if the food substance is not present in Jumo. So there's an example right here. So we have food nutrient reducing sugar, and the test is performed including all the reagents. So yeah, tell you how we're gonna do it. We mix Jumo with water and place the mixture in a test tube. Right, add an equal volume of Benedict's fillings to A and B solution, and then mix thoroughly while gently. Then the expected observation turn orange, right, yellow, brick red, and if it does not have reducing sugar, it will remain blue. So it's your time now. So
You understand, sir? That's a blind my eye.
so if it was starch, how would you test for starch here?
How you dealing?
Iodine starch test.
Right. So how would you go about doing that test? So follow the example at the top there. How would you go about preparing the mixture?
Um,
Sorry, I'll get the iodine and then
put it on the Juno.
spread it evenly on whichever thing them give, sir. So did it.
Then observe the color change if it's blue, black, or yeah, if it's blue, black,
Black or blue.
Hmm. Wait. Okay. Tell me what are you seeing on the screen? Oh, you're
Okay,
seeing the question, right?
yes,
Yes,
sir.
sir.
So the first thing you say you're going to do is we're gonna do with the Jumo for test. You're gonna spread the thing on the Jumo. That's what they said. You're gonna spread iodine on the Jumo. Yes?
So repeat that yourself.
All right. So the first aspect, right? I'm asking you what did you say if you're going to spread the iodine on the Jumo?
Yes, sir.
And that's the same thing you would do, recorder. Hello? Let me stop share the screen because
Yes,
I'm
sir.
not sure which of the screen is sharing. My bad.
It was the past paper.
Oh, because me have many screens up, that's why. All right, so you're seeing the paper now, right?
Yes, sir.
Okay, right. Something is not connecting right. I'm just trying to sort it out. Give me a minute. All right, so it should be back up. You hear me clearly?
Yes, sir.
All right, so the first thing we said that we're gonna do, we're gonna use iodine solution. All right, so that's step number one. Right? But what you're going to do with the iodine solution, you can't just say I go spread it on the Jumo. Remember, you don't remember when you're doing labs, you don't just spread things. What's the first thing you're gonna do when you're doing a lab like this? And if you follow the example at the top, what would you be doing? Because remember, guys, most of these questions are
Sure,
thinking
so you can
content.
put the iodine solution in the test tube.
Exactly.
Then you put whatever you put in it.
No, no, man. Tell me where you're putting it.
The
So the
Sir,
first
Juma.
whichever
thing So the first thing you're going to do, you can't just say whatever. Think about it like a lab, right?
the something necessary I put in it. The Joom or something.
Right. So the Juma is what? So think about it. All of you guys do labs, right? You must. As if you do labs, you wouldn't be here. Cool? So if you are going to do the test, you have to think about it like a lab. You have to do exactly what a lab would do. And the first thing you're going to do, you're going to mix the Juma with water, right? Because remember, the Juma is a powdery substance. Remember, he said that, right? He said up here that the Juma is a powdered food item. And they gave you an example right here. To say mix the Juma and place the mixture in a test tube. So you're gonna keep that same thing. So after you mix the Juma, no, step number one that you mix the Juma and put that in the test tube. What's step number two?
You add the iodine.
you add just iodine or you add a few drops. 'Cause remember say it is a
Yeah, the
drop.
drops with the dropper thing, sir.
You
It's a
drop.
droplet, right? So you say you add a few drops of iodine solution to the mixture. And what's the third thing you're gonna do?
You mix it.
You shake it gently or mix it. Right? So all that would be the first step. Right? Then now, what you're gonna do after that? What is the expected observation now?
See if it's going to turn black or blue, sir.
So
Blue
it's gonna
black.
turn blue-black. Right? So if it is present, then of course it's gonna turn blue-black. If it is not present, what's gonna happen?
The color remains.
No observable change.
Go again.
No
The
observable
color
change in color.
remains. Yes, I
Right.
wouldn't. With Esper, what's your real name again?
So,
You are which? What's your name?
Esper,
You are from?
St. Vincent. I'm the one who know message you a few minutes ago.
All right, good, good, good. Just
Yeah.
want to make sure. All
All right.
right. So make sure you participate, Esper. All right. So good. And then we will have you will say that it will remain the same or remain brown or yellow. But remain the same. The color remain the same. Cool? How about the next one where I'm gonna type this? How would you go about fat?
Mix the jumo
Emulsion test.
So how you going about that? So you mix it Jumo,
with water.
okay, good. And what else?
I think add ethanol.
Then you add ethanol to the mixture, and then what you do? Cause this one is kind of a little bit trickier, for fats. So for fat, you're gonna mix the Jumo, right? And then after you mix the Jumo, you're going to add ethanol to the mixture and you're gonna shake. Right? After you shake, what you gonna do after that?
Well, you make the observations and I guess you look out for cloudy suspension.
Yeah, but you not make the observation yet. Cause you have to do something before that. You would have to
Sir, put in
add
a test tube with boiling water.
you would have to throw back that mixture after you shake it. After you shake it, you go pour ethanol mixture into another test tube containing water, eh? We could do that. But I'm not sure if if you need to go that deep still. But okay, fine. Let's leave it at that. Then what's your observation? And then what's the observation if Juma is present?
Formation of a cloudy
Cloudy
suspension.
suspension.
So cloudy suspension, right? So we say milky white emulsion. And then now what is it if the fat if it's not present?
Well, no suspension forms or no emulsion forms.
So it would remain colorless. So there will be no milky white. It will remain transparent and clear. It won't be, yeah, transparent and clear. All right? And this
So
should give you
you could
some
use grease
marks.
spot test.
Yes, you could. You could use the grease test if you wanted.
Uh,
We know how to do that with the getting the paper and see if it spots.
Yes, sir.
Okay. So we have two tests for fats. And Mark are good at both, right?
Yes, sir.
All right, next one says stage 3 precaution. All right, that should be taken while conducting the test. All right, and the precautions are normally the same, but let's hear them. 3 precautions. Let's go with recorder. Give me have 4 people here. Yes. So let's get one from recorder. All right, Espa, in the interest of time, can I have a move?
I guess you could ensure that all apparatus is sterilized.
All right. Why?
I guess to ensure that no contamination occurs between the different tests.
Yes, because the contamination will cause a false result, right? So it can cause a false result. So it can tell you that it has it, but it didn't have it. All right? So that's good. What's the next one? Markel.
Oh,
Oh, yes, record. I'm here with Payola. Come on.
sorry. I was saying that we could use the in regards to quantity, we can't use too much because it can affect the outcome of the test.
Okay. All right. Who else is here? Belle, Sasha.
Sir, do not ingest the food after conducting the test on it.
Okay. Okay. All right. So yes, right? We do not ingest. We can all when we're practicing lab, we always do what? We put safety and we wear our protective gloves, not true, and our goggles
And or coats.
Or
and
don't
our
taste it.
coats.
Don't taste not taste the chemical.
Yes, so we wear our coats. That's why our scientists always in our coats and our goggles to protect us when doing um experiments. Right? Um and another one could be what? It could be we'll be heating, like heating equipments. So we ensure that the test tubes are not overheated. If the test tube are overheated, what would happen?
Correct.
They would crack or burn and the glass would break. Right? So I think that's pretty clear. That's pretty easy. Easy stuff. Name 2 diseases that a person may develop if Jumo is consumed in excess over a long period of time.
Diabetes,
Diabetes, excellent. And what else?
heart disease,
Sugar? Yes, it
Atherosclerosis.
could. Huh?
Atherosclerosis
Sure,
in the arteries.
sure. Think about it. It's a sugar substance. It wouldn't necessarily be a fat substance.
Oh, I thought Juma had fat in it.
No. So Juma is a so them say it's a powdered food item, right? It includes starch, reducing sugar. All right, fats. Yeah, it could, It could. It could be correct. Yes, it said fat in it. So yes, I could accept that. Yes. You're right, my youth. Indeed. So obesity, anything we have to do with fat, and you could also have obesity. And what comes from obesity? That could be an easy
Heart
one too.
disease.
Yes, and which one type of diabetes would you normally get here, sir?
Type 2.
Yes, type 2 diabetes. All right?
So that's when the body doesn't produce enough insulin, or is that the one where the cells become resistant to insulin? Which one is it?
Which one of them, guys? For type 2?
Insulin resistance.
Yes, yes. Good now?
Yes, sir.
Yes,
All
sir.
right. So, next question says, which structure this is shows a simple diagram of a Villus in a in the digestive system. Right? So we skip ahead, name the organ in which the Villus is located. What's
Small
up?
intestine.
Small intestine. And we know the function of the Villus.
Yes, sir.
What is that?
Increase surface area for nutrient absorption.
To increase surface area for nutrients absorption. Everybody agree with that?
Yes, sir.
All right. And name the structure labeled in X. X.
So would that be lacteal, or is that lacteal?
Lacteal. Lacteal, yes. Lacteal. Eh? Are we comfortable? For that's one thing though. Do we know diagrams well?
Yes, sir.
Yes, sir.
Yes, sir.
Michael? You know your diagrams well?
Sir, when I get much of that,
So you have to know your diagrams. I have a video there that kind of capture all the diagrams. I'm going to send it to you. You can not do by and know your diagrams, right? You need all of your diagrams. You need to know all of them. You need to know how to draw them, and you need to know how to identify them. All right? You know the Vilnius, right? Michael?
no, sir.
All right. So the Vilnius increases the surface area for absorption of nutrients from digestive food in the bloodstream, right? Then name the structure. You guys said it was lacteal, right? So ensure that you know your diagrams well. Next, it says John discovered that the structure labeled X in his digestive system is not working properly. So just one adjustment that he should make to his diet because of this malfunction.
Sir,
Eat
remove
less fats.
fats from his diet.
Yeah.
Excellent. Excellent. All right, so he should reduce his intake in fat. Why?
So
Because
because
the lacteal is responsible for absorbing fats, and if it's not working properly, that means fat is not being absorbed. So you should just stop consuming fats.
All right. And also, it is responsible for fat-soluble vitamins as well.
Mhm.
All right. So indeed, excellent. Recorder, you good?
Yes, sir.
If you're not comfortable with the answer given and you want me to elaborate on it, please say so.
Yes, please.
You want me to elaborate on it?
Yes, sir.
Which one?
The bottom one.
The lacteal? All right. So it says that John discovered that the structure labeled X in his digestive system is not working. Right? So just one adjustment he should make to his diet because of this malfunction. So the lacteal is not working. Right? When the lacteal is working, its main function is to absorb fats. right? And fat-soluble vitamins. Do we know what fat-soluble vitamins are?
So I just
Talk to
hear the
me.
term and use it. I don't really know exactly what it means.
Huh?
I hear the term and I use it in the exams, but I don't really know exactly what it is. I just
All
assume
right,
it's
so
vitamins dissolved.
fat-soluble vit all right, so fat-soluble vitamins are vitamins A, D, E, and K. All right? They are A, D, E, and K. Right? And they are stored in the body's fat tissue. So they are stored in the fat tissue. Right? That's what fat-soluble vitamins are. And the lactate is responsible for absorbing fat and absorbing those vitamins as well. So in order for your body to absorb vitamin A, D, E, and K, lactate has to be working so efficiently. Right? So if your lacteals are not working efficiently, it would mean that if you are consuming fat, then the fat will not be absorbed. And if the fat is not absorbed, what will happen if fat is not absorbed in your body?
So, well, I guess you lose the benefits of having fat consumption, such as increased insulation and that type of stuff.
If the body is not all right, what else? That's a good answer. What else? Anybody else? No? So if fat is not absorbed in the body, well, it causes damages to our liver tissues. That's for one, right? And the fat will stay in the bloodstream, and that's not good, all right? That's definitely not good. So it damages your liver tissue, right? And some cancer can cause from it, but not all. Some cancer. All right. But in addition to that, it directly damages small intestine, pancreatic or liver tissues, right? And other conditions that you can slow down digestion.
So,
All right.
yes, sir. I have a question.
Good. Go ahead.
So, is atherosclerosis, that's when you have a buildup of fat in your arteries. Would that be
Mhm.
caused by absorbing too much fat or not absorbing enough?
Can be. It can. It can be one of the reasons. It's not most diseases come from more than one reasons, right? And this can be one. Understand?
Yes, sir.
All right. So if the lactate is not working properly, then we would have to slow down the amount of fat that we take in because the body then will have too much fat. Understand?
Yes, sir.
So,
Great. So good now, recorder.
Yes, sir.
All right, next question says, give one reason for your answer in E. What is 4? Is this E? Oh, yes, it's E. Oh, yes. Give one reason for it, even though we kind of get a reason already. But anyways, give one reason for E4.
So like you said, the lacteal is responsible for absorption of fat and fat-soluble vitamins. So when it's
Mhm.
not present, you have a buildup of fat which could damage small intestines.
Good. Excellent. Everybody Gucci?
Yes, sir.
Yes, sir.
Right. Next question says to give one reason for figure 2 shows the epithelial cell of the Villus in the digestive system. What is an epithelial cell though?
I think that's one cell thick.
Hmm. Anybody else? That's not wrong though. Yes, it is. Anybody else with anything else you want to add to that? Want to hear that word, epithelial. What comes to mind?
So if is a guess, but epidermis comes to mind.
Mm-hmm.
So I maybe epithelial cell would have a cell wall.
Mm. But what does epi mean in biology? Because remember in biology it's all about prefix and suffix. So
Outside.
even if you lose it, right? Even if you lose it and you don't know it, you can work it out, right? So epi means surface area, right? That's why epidermis is on the outside, on the surface, right?
Yes,
So,
sir.
right, so it's one cell thick, it's on the surface, right? So outline one way in which the epithelial cell of the Vilnos differ from a typical animal cell.
So well you could just say the
Mm-hmm.
epithelial cell of the villus is armed with lacteal, while a typical animal cell is not. I don't know.
Mm. No, I'm reason, me rather reason it out me rather reason it out than than just stay quiet. All right, good. So you said that the repeat what you say, and before you repeat, is it Jordan or Dixon? Yes, I'm seeing your responses, sir. Right, and if you're interested in joining us, you can also send me a WhatsApp message and we'll discuss it. All right, so anyone on YouTube interested in joining us because this class will be ended in the halfway segment because of, won't be doing the full paper here. All right. So you said, repeat what you said, Espa.
I said that the epithelial cell of the villus is armed with lacteal, while a typical animal cell is not.
Hmm. Okay. Anybody else? Recorder? If I look
Um,
on it, if I look on it, it gives it away still,
it's
I will
shaped.
take I will take that answer though, Espa. That answer is correct. Yes, recorder, give me another one.
Sir, I don't know. I was just about to say it's shape, but like, never mind.
It's right there though. It's right there. Sasha,
Sir, the microvilli
huh?
different. Is it that the
Yes,
epithelial,
yes.
I don't know how to pronounce it. That's of the
Microvilli.
microvilli, and then the animal cell doesn't have one.
Yes, there's a microvilli, the finger-like projections of the Vilnos. We don't know about the finger-like projections? No? The finger-like projection, the microvilli, has finger-like projection. And what's the purpose of the finger-like projections?
Increased nutrient absorption.
Yes, an increased surface area, right? Remember, you can't go wrong with that. increases the surface area. Right? So remember, we are asking you about, it says outline one way which is the epithelial cell. So the epithelial cell differ from a typical animal cell, right? And that is that it has a vilnus. It has a microvilli, right? Sorry, not vilnus, microvilli, that finger-like projection on its surface. It greatly increases the surface area for absorption. Right? Good with that?
Yes, sir.
Could you give another way? Could you give two more ways in which there's difference?
So I know the villi, they're also equipped with capillaries, and maybe not all typical animal cells would be.
Capillaries. Not bad, you know. Not bad. What else? Remember, guys, that first of all, in order for the microvilli transport, the vilnus transport food and nutrients. Not true? Is it true or lie?
True, sir. True.
Yes, Trish.
And if it transfer food, right? What process would it transfer nutrients in?
Oh, diffusion.
Think about
Oh,
it.
wait, what?
Think about it, not diffusion. Because these molecules are bigger.
So active
Nutrients,
transport, maybe.
active transport. And what does active transport need?
Energy.
And what gives energy? What gives energy in any cell or any
Mitochondria.
structure? Mitochondria. So therefore, because of that, you would need a high the Vilnus would have a high concentration of mitochondria to facilitate the transport of food and nutrients. Make sense?
Yeah, so
And also an easy one you could say is that it's adapted for absorption. You know, my animal cells are not adapted for absorption. So you find all the functions of the Vilnus and you can put it there. Right, so I'll give you one more. High mitochondria concentration, right? And of course, lacteal. No lacteal there. So there are many different answers you can give. Jordan says, so how do you feel that regional integration did not come on Sochi? It was expected. It was expected because it's a new syllabus and them give regional integration every year. So it kind of was expected, but we're still prepared for everything. All right, next says, name the structure in figure 2 which carries genetic information in the form of DNA.
nucleus.
All right, nucleus. Everybody agree with that? Or we have something different?
Yes, sir. We agree.
Yes, sir.
Nucleus.
All right, good. Then we have my favorite part of the test. Our graphs. So let's look at it. We say figure 3 shows the effect of temperature on the rate of an enzyme catalyzed reaction, right? One I indicate by placing letter A on the graph in figure 3, the point where optimal temperature of the enzyme catalyst reaction occurs. Right? So it says by placing letter A on the graph, the point where optimal temperature is catalyzed, right? They can to utilize the animation, what is this? The annotate and zoom, they can do that for me. Oh, man, I see. Somebody's doing it.
Yes, Esper just did it.
So Esper says letter A points where optimal temperature of this graph catalyze reaction, right? You believe that they think that is correct?
Yes, sir.
And what do we mean by optimal temperature?
The highest.
Right? The highest. Put some more explanation on that. For the man them when I understand what about.
So the optimum temperature is where the enzyme would function at its best rate.
Yes. So it is the point where the catalyzing of the reaction goes the best. Right? And we said that enzyme is anything that speeds up a chemical reaction, or catalyst is anything that speeds up a chemical reaction. So this is optimum point where it is the fastest. Right? Then it says state the optimal temperature of the enzyme. And what is that?
40
This
degrees
is
Celsius.
40. 40. How would we do this if we are in the test?
Put a ruler on it.
Oh, yes. Beautiful. Put a ruler on it because you don't want to make that mistake, right? So you put a ruler on it just to make sure. So about 40 is indeed correct. I see some people 38. That's why you put a ruler on it, right? It's 40. Cool?
Yes,
Then
sir.
we go to the next question. Oh, sorry, man, we was the wrong thing here, so my bad. Yeah, we know say 40. I move because of time. Then it says, explain the shape of the graph between 0 and 25 and between 50 and 70. Okay. So I'm going to explain between 0 and 25 first. We want to explain between 0 and 25.
So between 0 and 25 degrees Celsius, the rate of reaction is increasing at a steady rate. Well, yeah, it has a pretty steep gradient. That's about it.
Mm. Anybody else? I feel that that's it. There are a few lines here, you know. Espas, a few lines now only means there are more than just one sentence, eh?
Yeah, so I mean, if it gave more information, generally you could say the increased temperature results in increased kinetic energy, but they didn't really give a lot of information for you to work with.
And that's the thing. When them give a little bit of information, you have to use what would normally happen. Right? So why the first obvious thing here is what Mr. Esper said. And Esper said that let me go back here. He said that the reaction rate increases gradually. That's what he said, eh?
So the higher the temperature,
Yeah.
the faster the rate of the reaction.
Higher the
Which,
temperature. Right, because it is temperature dependent.
yes.
Exactly. So we add that. So we can say, all right, then it's temperature dependent. So we see how it is temperature dependent.
Yes, sir.
Everybody else here is temperature dependent. Sasha, Espa, Bell.
Yes, sir.
Yes, sir.
If you wanna see it, you
Yes,
tell
sir.
me, Tell me. Tell me. Don't hold it. Alright? If me I use a word, we seem bigger than you, tell me. Right? So we would say that it's temperature dependent because the higher the temperature, the faster the reaction. So that's the first obvious thing. Then now, why would it be temperature dependent? Because normally when you get these graphs, right, you would have to, after you make that observation, the next reason normally why would it be like this? So why would it be that? You know? I don't understand what I say.
So why would
So
it be
now,
what?
all right, so normally you said you are the one, the normally, all right. So normally the observation that you make, right? And this in this instance, the observation is that between 0 and 25 degrees Celsius, right? The there is a steady increase. The reaction rate increases gradually, right? Because of because the temperature also increases, right? All right, it's temperature dependent. We agree to that, right?
Yes, sir.
Yes, sir.
Now I'm saying to you, why is the next step that the next step that you would take in answering this question is answering why it would be increasing, right?
Oh,
Right. So you'll have to explain that why it was lower at 5 degrees, why it was higher at 10 degrees, why it continues to climb. That is your next step. So even though you don't see it on the graph, they expect you to tell them what is your next step. So remember, we are not here to swat questions only. We are here to understand the concept of how to get maximum marks from these questions. Right? Every year, my students get ones. Every year. No joke. You can check for yourself. And they get one because of understanding how answer the question. It's not necessarily just to swap the question, right? But to answer the question. Because you'll never see questions directly back, but you'll see them in a way where if you answer one question, like if you answer this, you can answer another one like this. You get where I come from? So
Yes, sir.
the next step we would do here, let me explain. Now, we will say that at lower temperature, the enzymes molecule have a low kinetic energy, right? And we know that when enzymes work fast, it's because that the collision between them is fast. Do we know what collision means?
yes, sir.
Yes, sir.
So faster collision means that they are moving faster to break down what they need to break down. True? So
Yes, sir.
we would then say now that the reaction rate increases gradually because the enzyme activity is temperature dependent, or the enzyme activity increases as the temperature increases. Then we would say that at low temperature, right, the energy molecules have low kinetic energy, leading to fewer successful collision between the enzymes and substrate molecule. I want to know what is a substrate, right?
Yes, sir.
Everybody know what's a substrate.
No, sir. Could you please
Yes, sir.
say what it
So
is?
a substrate is the food that is being broken down. Right? So the instead you say chicken, or if you have starch in your mouth, all right, let's say for example you eat rice, you would not say rice. You would refer to the rice as a substrate. So the enzyme binds to the substrate.
Okay, sir.
And where does the enzyme bind to the substrate? Just for a rule's sake. What's the place? Where is this place specifically where the enzyme binds to the substrate? Hmm? We don't know?
This is like the lock and key mechanism thing.
Yeah,
So like that's
that's
the
the
active side.
process. The active site. That's the place. So where the substrate binds with the enzyme, it is called the active site. You understand that, recorder?
Yes, sir, I understand.
I will know what is lock and key, recorder.
What?
Lock
No,
and key.
no, sir.
So at the active site, you know different types of enzymes. So keep your mic on, please. Let's keep this interactive. Recorder, let's go. So you know, right, the different type of enzymes. Tell me them. Tell me two quick and fast.
What did you say, sir?
Two different types of enzyme. Tell me two different type of enzyme.
Um, um, lipase.
Hmm. All right, let's help record out. Yes, that is one, but you don't seem confident. Give me two different type of enzyme. Let's start where it all starts. And YouTube, I'm
Trypsin
watching YouTube. Yes. Where does digestion begins?
in the mouth.
So the
And what's
more
the first enzyme that's there?
salivary amylase,
The amylase. And what does amylase break down?
starch, carbohydrates.
Starch. Did we know that, recorder?
Yes, sir.
Next, we're moving to the stomach. Which enzyme is in the stomach?
Pepsin.
Pepsin.
Pepsin. Right? So why can't amylase and and what does pepsin break down?
Proteins.
Protein. So why can't amylase break down protein?
Oh, that's because it has a specific substrate. And yeah, something like that. And protein
Hmm.
does not fit.
Yeah, you kind of get it when I explain it wrong. Want it. You see me? No,
Yeah, I understand.
it's the same concept like what lock and key is. Recorder,
Yes,
if
sir.
can I use my key to open your door?
No, sir.
No. So that's what lock and key is. All enzyme can only lock into a substrate that is meant to break down. So for example, if amylase is a triangular shape enzyme, it has to break down starch because starch is also triangular shape and it can fit at the active site. And when it can fit, it locks together to break down the substrate. If enzymes try if pepsin is can only break down protein because it and protein has the same shape and they can fit. That's what the concept of lock and key is all about. So if amylase tries to break down protein, can a triangle fit in a square?
No, sir.
Nope.
And that is why enzymes are specific. That is that is what lock and key means at the
Okay.
active site.
Okay, sir.
Is that clear?
Yes, sir.
So all who never understand that, you're supposed to good now. So back to the question. So we are saying then, we understand how we approach the question so far. We understand this answer.
Yes, sir.
Okay. So then we will say, as the temperature increases towards 25, what happens there as it moves to 25?
Increase kinetic energy, so you have more collisions, more effective
Oh
collisions going
God,
on.
yes, sir. Yes, sir. Yes, sir. Yes, sir. Excellent. Excellent.
So you could relate the whole thing to the collision theory of matter from chem.
Yes, you can, because chem and all the sciences are related as you move up in a life. Now, can we now attempt 50 to 70, please? Between 50 and 70, what's going on there?
There
The
was a
enzyme is
decrease
being
in
denatured.
enzyme activity.
So there's a decrease in enzyme activity. Why? Let me see who can put the let me see who can put the best string answer
You have
together.
when the temperature is too high, you have denaturing of the enzyme.
So put that in one sentence now.
So I guess you could say as the temperature increases to a certain extent, which would be between 50 and 70, the enzyme begins to denature. So I guess you have a few amount of enzymes for effective collisions to occur.
Sir, and could you say that because it reaches its optimum, that's the reason for its denaturing?
All right. When it reaches you could. However, ESPA answer is more spot on because ESPA answer, all right, both answers are correct, but one answer gives specific to some extent. Why?
No, sir. I'm saying like that could be added to his answer.
Okay, get you now. Get you now. Good. The only thing where ESPA miss is what? You know, the only thing we miss ESPA in that wonderful answer?
Not sure.
All knowing that tell me say the reaction decreases.
So,
You never tell me.
so repeat again, please.
You did not state that the reaction would decrease.
Oh, I go around it.
All right, you said, yeah, you said that the temperature increases, right? Which causes the enzyme to denature denature because the temperature increases, right? That's essence of what you said. But you never say, then you said the collision would it affects the collision rate, and then that was all fine. But you could have said no, which led to the reaction rate decreasing. Because that is what is happening. That's the first thing. Let's look at the diagram. That's the first thing that's happening. You have to acknowledge the decrease. When you're looking at a graph, you acknowledge what happens first, and then you tell us what is happening. Why? So look at this first question. The first thing that we said that the reaction rate increases. Can we see it though? And then we went ahead and said, why did it increase? You get where I come from?
Yes, sir.
All right. Recorder, Sasha.
Yes, sir.
All right. So we start by saying the reaction rate decreases, right, sharply because the enzyme begin to denature at high temperature. I want to say that we're good. Right? Anything else around that is fine, as long as we have that the reaction rate decreases. And because it after reach optimum temperature. Alright? What else could we add there?
So same thing I said about less enzymes available for effective collisions.
Alright, we could add that too. But we need to expound now on denaturing, because that is the main reason why it's happening there. Denaturing. What is denaturing?
Basically, when the enzymes start to shrivel up or die,
No, you know. That is the biggest misconception in biology. Trust me. Every year
So
it chop kids.
they lose
This
their function.
it loses what?
Its function.
Its function. All right. Before me address that, may I just tell us that this question, denaturing, chop at least for 80% of CXE kids every year. So don't make it chop, you guys. All right. Denaturing, right, is the slow down of a chemical reaction. I could say it again. An enzyme never stops. It slows down. I say it again. Enzyme never stops. It does what? It slows down. Is either it go fast or it go extremely slow. It got so slow that it now work out. Hmm? But it never stop. Right? So that means that why does it slow down, right? Because when it slowed down, what happened is that what is happening why it slowed down? I could see who have that.
so the more
Jordan.
possibility. Oh,
Yes, Jordan, you're absolutely correct. Go ahead, Esper.
so I was saying, as they slow down, there's a lesser possibility of collisions.
Think about denaturing. What is denaturing? Why does it slow down? Because the lock, the lock chart start to change. Make we pray. So we go back over here, so, right? We're going to use amylase as a triangle, right? As soon as it start to denature, the shape of the triangle start to change a little bit. So it start to change. So it's no longer this anymore. It start get smaller. You get what I say? And if this start to change, it means that it can bind with the substrate well enough. So because it can bind with the substrate well enough, only piece of it can bind, only a little piece of it can go in. It's not going in all the way. It means that the chemical reaction is being slowed down. Does that make sense? I will lose somebody.
It makes sense, sir.
We lose somebody so far? All right. So we understand Espa.
Yeah, so
Sasha, Ziggy.
Yes,
Yes,
sir.
sir.
All
Understand.
right. So we are gonna add that. So we are gonna say that denaturing this is why it is slowing down, guys, because denaturing is a process where enzymes active site loses its specific shape, preventing substrate binding. You said that you give that answer on your paper, you're wicked. Tough. Tough. And that will give you full 4 marks. Are we comfortable?
Yes, sir.
All right.
Yes, sir.
Let's check what's going on. Them said two scientists visited a rainforest and noticed, right? So two scientists visit a rainforest and notice orchids growing on a large, healthy tree. Name the type of relationship that exists here.
So maybe commensalism.
The commensalism. The group agree?
Yes, sir.
You sure?
Yep.
So put me tell you,
Sir.
Sierra.
Sir, I'm very confident that commensalism is the answer.
I said put me tell you, Sierra.
Then me know what happened there.
So if not commensalism, maybe mutualism. If
I'm
not
just, I'm just joshing you guys. It's commensalism. I want to see if you are confident in that answer. Yes, it's indeed commensalism. Then it says outline. Do you know why this commensalism before me chop on?
yeah, so
Yes, sir.
Right? So both organisms benefit from each other. And none is neither help nor harm, right? So both of them living on each other. It can't be mutualism because mutualism means that both of them would have been getting source of nutrients from each other, right? So yes. Next one says, outline two reasons for your answer. I kind of give you one already, but let's go.
Sir, the tree is not harmed.
Neither of them is harmed.
So the tree is not harmed. So the tree is not harmed nor benefit from the orchid presence. Alright? True?
yeah,
Excellent.
so
Yes, sir.
Then we say that what's another reason we could put there?
the orchid benefits from the tree's presence because it provides structure, and I guess it could get nutrients and water from the tree.
I like that. So the orchid actually a gain from the tree. When I say the tree not gaining from the orchid, But the tree is not harmed by the orchid either. It's not like say the orchid have some root in the phloem and a thief its nutrients. It's not. But the orchid is being benefit from the tree because of the height to get better access to sunlight, as a great strawberry said on YouTube. And why when it's high, what happens when it's high? It won't be competing with no other plants. You see that? You understand that?
Yes, sir.
Yeah, so
That paper look easy. I will find one tougher paper. This says, name two animals that are involved in a type of relationship named in AI.
so the relationship between a cattle egret and a cow,
The relationship between a who?
a cattle egret and a cow.
Oh, yeah. Explain that. Because the cow is not harmed by the eagle, but the eagle eat off the ticks off him. That's where you're going, right?
Yeah, and the flies. Yeah.
And the flies. Excellent. The cow and the egret is the most common one. Them ask that for every paper. If you've been looking at any paper. Right? And then what else? Can you give me another relationship?
So
They
it
said,
had one with a shark and some small fish, but I can't remember the name.
yes, they call them the remora fish. Right? So it says two animals that are involved, the cow and the egret will be good enough. But since we always, since we know the cow and the egret, we are gonna just drop two things on it for you people them who want extra and seem like them brighter than everybody else. You understand? So we are gonna say the barnacles and the whales. That's what I said. And then we are gonna use the remora fish and the shark. Because we're active over here and we give big things and we like give big things, make people see so we're at. I say. So let's move on. Next one says, the scientists later found an unknown. Sorry guys, sinus was acting up. So the scientists later found an unknown organism in a body of water. They were undecided about whether it was an amphibian or a fish. So just the structural features of the organism may be used by the scientists to determine its identity. And your answer should include two comparisons. That one you look rurish. But you want to answer this one now. See how we take on that one. So the scientists found an unknown organism in the water and they were undecided about it, whether it was an amphibian or a fish. And that's easy. Suggest two structural features of the organism that may be used to determine. So what's the difference between an amphibian and a fish? That's simple something then. Simple something.
So
Roll
the
up.
skin,
So what we say?
the the skin of the reptile has dry, scaly skin, while the amphibian would have moist, wet skin, moist, smooth skin.
Yes. Why does the amphibian have moist, smooth, slimy, wet skin? See if you're active. Let me see if you're active, Esper. And I'm going to go up on YouTube, you know. I don't want a YouTube answer. I don't want a Google answer, I should
Why?
say.
So
Yes,
why they
why?
have smooth skin?
Yes, why they have smooth, moist skin?
Um,
If you tell me why I'm fly go to St. Vincent when summer come. Angry are you?
maybe why they have smooth, I guess maybe they could have the skin could be involved in some sort of osmosis process, maybe,
Mm,
or diffusion, because that's a feature of a
mm,
diffusion surface.
You watch cricket? You watch cricket, Esper?
So,
You watch cricket?
nope.
Mm-mm. I mean, so we can't give a West Indies ticket them for. Alright. Why is this? Why the skin? We know that water is very important, right? When it comes on to respiration and gaseous exchange. And that is why an amphibian skin is moist. Because an amphibian breathes through their skin. Hmm? We
Not
want
the
know
same
that.
diffusion thing I say.
They're close, but they're never close.
Yeah. Well, they're not aspirate anyway, so same thing.
Right? No. What we are doing here, you know, if you notice, me I always ask you follow-up questions. I
Yeah,
ask you
so
follow-up
understand.
questions, right? That if they ask this question another way, you still have it. Hmm? And to have a better understanding. Right? So yes, that is why. So that is one reason, the skin. One has scaly skin, right? And one has smooth skin. What is another one? Easy.
So, Fish only lives in water while amphibian lives both on land and water.
But you're missing one more thing for buying up that now. Buying up that now, Ziggy Marley. Buying up that. If one live on water and one live in both habitats, what would one have where the other not have?
Legs.
Breathing mechanism.
I understand where you come from, but no, not in this context.
So the breathing mechanism.
What is the breathing mechanism?
The fish should have gills.
Huh?
The fish should have gills.
And the amphibian would have what?
Sure, they'll be skin.
Oh man, chop yourself, man. Come on, man. Come on, man. Ziggy, man. Ziggy James, man. Ziggy, you say one live on land and water. One live in a water. And then me I ask you now, based on that, what one of them would have what the other one not have? I'm not talking about legs.
So, tail, like for swimming, or features for swimming.
Ziggy, where Aquaman have what we not have? What does Aquaman have that the fish doesn't have?
Sure, foot.
Tell me foot, boy. Before we compete, your money. Let you tell me foot. Hey, put my foot in that. Well, better man, me take your amphibian foot and kick it out Texas. Lungs, Ziggy, lungs.
Yes,
We see Mickey on it on YouTube. We see Mickey with it on YouTube. And we see Cyrus after many guesses on YouTube. Come on, guys, lungs. An amphibian lives on both land and water because an amphibian has both gills and lungs. They have two breathing mechanisms. While fish can breathe outside of water because they only have gills. Huh?
yes, sir.
Ziggy, you don't think you forget a beating?
No, not yet.
Ziggy, you forget a beating. All right, people on YouTube. We can't believe that this a chop on, brother. This can't chop on because you have to think critically when you reach the exam. You get what I say? You have to rethink critically.
Yes, sir.
Yeah, what I say? So fish has skin, scaly skin, and fish only have gills. Right? So them not have lungs, so them can't breathe outside of that. So them two things there could differentiate between the two. That's all. We
Yes,
all
sir.
understand that? We understand that?
Yes, sir.
All right. I remember that just to clear it up for being specifically, right? Just to be specific. An amphibian breathe through their skin and their lungs. But a young amphibian, like tadpole, have gills. But a big amphibian would not really have gills. They would have skin and lungs, just to clear it up for everybody. So I'm going to put it in the answer just to clear it up, because I don't want nobody go say amphibian have gills. Only young amphibian has gills. We clear? Are we clear?
Yes, sir.
Hakeem James said big up Ziggy. Cause you make a valiant effort. Alright? Bell, Shabir Bell. Are you good? Little quiet one, I have to ask them all the while, you know. I have to ask one all the while. Alright, this says define the term decomposers. Now that's simple. Go ahead and do that.
So decompose is an organism which feeds on dead matter for its functions, I guess.
So decomposer feeds on dead matter. You know, I'm not gonna give you that Esper. I got tougher, I'm not gonna give you that. That sound like a saprophyte. I'm not giving you that one. Don't think better than that, Esper.
So, well, if you asking me to distinguish between it and like a saprophyte, I guess you could specify on the dead matter, you could include plants as well, plants, animals, waste.
All right, watch it. Watch it now, no? Give me some examples of decomposers.
Maybe a vulture, or that's not I don't vulture.
And I just say you go wrong now. And you say that's why I asked you that. A decomposer of bacteria and fungi. You know what I say?
Yeah.
Because a decomposer breaks down dead organic matter into simpler matter and release back the nutrients in the environment. You see that?
Yeah.
That's the big difference. That's what a decomposer is. So a decomposer is an organism, right, such as a bacteria or fungi, that breaks down dead organic matter into simpler substance, releasing back the nutrients back into the soil or into the environment. Make sense?
Yes, sir.
Yes, sir.
All right.
So
Then-
what's the difference between like a decomposer and a detritivore and saprophyte?
So you're asking now, what is the difference between?
Decomposers and detrivores and saprophytes, them type of thing.
All right, so tell me what is a detrivore?
I really can't remember now. That's why I asking.
All right. Think about it. Vores. What does vor mean? Carnivore, omnivore. What does vor mean? That's why I say prefix and suffix is biology and you're good.
So maybe like a feeding relation, I guess.
So detrivore. Vores is something that eats something. Consume it. You understand that?
Yes, sir.
So a detrivore is an animal that consumes dead things. Eat it. Right? So a detrivore could be decomposers. You understand that?
Yes, sir.
Because in breaking it down, they would also be eating it as well. Right? And a saprophyte, an organism that obtains, what do such saprophytes do?
So if I try to define it, does going to sound the same as a decomposer because
It
it's
kind
organisms
of
that
is,
feed on dead stuff.
It
Yeah,
kind of
that's
is,
the problem.
It is, All of them are related, but it's just in the context in which they are used. So they are overlapped, but it just depends on the context. Let me ask you for a decomposer. You can't just tell me a decomposer is an organism that eat organic matter. Because a decomposer actually releases it back into the environment. Even though a decomposer is a detrivore, because detrivore eat dead things. But if I ask you what is a detrivore, you don't say a detrivore releases it back, because not all of them do. You understand what I'm saying?
Yeah, so
So even though they overlap, right? You have to tell me specifically what me ask for. So me ask you a detrivore, yes, a detrivore is an animal that consumes dead or organic matter, right? It may ask you for what a saprophyte. A saprophyte decompose organic matter, and a saprophyte also, obtain nutrients, right? So what a saprophyte does is a saprophyte secretes enzyme to break down organic matter externally and then absorb the nutrients. That's what a saprophyte does. So a saprophyte, you see like how we have our enzymes on the inside, saprophytes have their enzymes on the outside. I lose nobody. I
that
lose
would
nobody
be
telling me, I not lose nobody,
no, sir.
right? So the detrivore, so the saprophytes, sorry, have enzymes on the outside, and they break down the material on the outside, the organic matter, and then absorb the nutrients afterwards. So technically, they are the same thing. They overlap, but they have some little tweaks when you're answering them individually. All right. So, what are two functions of decomposers?
So you just stated one before. Well, so recycling nutrients in the environment.
Yes, so they recycle nutrients in the environment or the ecosystem or anywhere that you want to choose. And what is another one?
So you could probably relate it to soil quality and its
Soil quality.
fertility.
So improve soil quality. Hmm? Or maintain, could say maintain and improve soil quality. It's just 2 marks, so that's fine. Good?
Yes,
Yeah,
sir.
so
All right. Next question. Run up in a say, outline one way in which fungi can be beneficial to a homeowner or a builder of wooden homes, and one way they can be destructive.
I don't see no benefit of having fungi in your home. So I don't know about that one.
So tell me that one way you will see.
So one benefit,
Mm-hmm.
I don't know. Like I genuinely don't know, to be honest. I don't know this one.
All right, somebody will know. I want to try. Sasha, now you're from a long time. Oh, Sasha. Oh,
There, I'm thinking.
Sasha.
I'm thinking.
Oh, Sasha, oh, Sasha, oh, Sasha. Oh, where is Sasha? Where is Sasha?
Sure.
Yes.
A benefit would be the fungi can help with insulation as it helps to bind the wood fiber to insulation boards.
Go again, sorry.
The fungi can help the wood fiber, help to bind wood fiber into insulation boards.
All right. And what is a disadvantage?
It can cause the wood to decay by
Mm.
breaking cell walls.
That's not bad, Sasha. That's not bad. So let's talk about the benefit. Fungi can help the homeowner to turn organic waste into compost, right? So the fungi will break down the organic matter and then return back the nutrients into the soil. So that could be a nutrients-rich soil where the homeowner can use for farming. Can put in a garden, make a nice little patio, nice little sweet garden. And so,
So that's why I'm going to ask if you could just, like, as a last resort, if you genuinely don't know, you could go back and choose one of the functions that you gave of decompose and say it could decompose organic waste and improve soil quality in the home. Could we
mhm,
set that up there?
mhm. What would be a disadvantage? Of course, Sasha said it beautifully. Sasha said that fungi can cause wood rot in wooden homes, right? Wreck the structure of the yard, you have to call man for repair it, and all of these things. Not true?
Yes, sir.
All right. So that make sense? That makes sense. All right, moving on. So YouTube, we soon close off because we're just doing half of the session to you guys. So if you want to join, you know where to hit the number below. And you can join our IT class coming up, bio class tomorrow. Yeah, we're fully active. Because we don't know why they decide to put IT and bio for the same day. That's some crazy
Sorry,
thing.
doing
Sir,
physics?
so you're doing physics?
Oh my God. After IT and bio, I don't know if we can match this. We can try that. Physics are like Tuesday, right?
Wednesday.
Sir,
Ah,
Wednesday.
probably me have a little time, can look at physics.
All
Mm.
right, good.
We can do so much things. Be like me, guys. Can do everything. All right. So easy question. Write a balanced chemical equation for photosynthesis, including conditions. Easy. You see this on the paper, you start to laugh. You take a breather. And you say, all right, CXC, do not insult my intelligence. Please, give me harder questions than these. Please. So let's go. Ziggy.
So,
Where's Ziggy there? What on the Espa? Where's Ziggy? Ziggy!
Yes, sir.
Come on, oh man.
Sir, carbon dioxide plus water.
Huh?
In sir, carbon dioxide plus water.
No, man, we want the
Plus
chemical equation, sir.
oh 1 second, sir.
Ziggy, you move so Ziggy. Ziggy,
Sir,
Ziggy,
yes,
don't
sir.
run. Ziggy, no, Carter, so I said, don't run. Don't run. Be the last man standing, Ziggy. What's the first thing that's going to be there in a chemical reaction for photosynthesis?
Sir, carbon dioxide.
And what is the equation for carbon dioxide? You know physics, Ziggy? You do chemistry, Ziggy?
Yes, sir.
Come on, Ziggy, man. That means that yesterday. How was it yesterday, by the way?
So it wasn't too bad.
All right. So, Ziggy, you must know the formula for carbon dioxide, Ziggy. Come on, man. You know chemistry.
Sir, CO2 plus
So, Ziggy, you make it look so.
H2. Sir, CO2
You
plus
have to
H2.
balance the equation, Ziggy. So, it have to be 6 CO2.
Yeah,
It's a balanced
plus
equation. Plus what?
6 H2.
6H2. Then you have the
Yeah,
variables, which are the conditions.
yes, sir.
So what
Then
are the
get,
conditions?
sir, sunlight. Yeah,
Light, Ziggy, light. Sunlight,
yes, yes, sir. Like, and then they
light.
get
No, you
C6H12
don't reach us
O6.
yet. You don't reach us yet,
Look at
Zigos.
that. Yeah, yeah.
Light over chlorophyll. No, so the green
Yes,
substance.
sir.
Come on,
Yeah,
Ziggy
yeah.
Boss, man. Ziggy Boss, man. You got the money
Sorry,
now?
it's slipping. It's slipping.
No, man. Boy, you're drinking rum.
Nah.
Ziggy, man. Drink some H2O, man. Let's go. So you
Yes,
have
sir. Then
6 CO2 plus 6 H2O
to
light over
over
chlorophyll
chlorophyll,
and the little
and
arrow
then
there,
you get
so.
yeah, and then you get
Uh, mm,
C6, and I think it's H12
mm-hmm,
and O6
mm-hmm.
plus 6O2.
Yeah, come on, Ziggy, man. Good, man.
Yeah, yes, sir.
All right. Think me can type this out, can I? Do I really need to type this out? I don't think so. So I'm going to try cut and paste from the Word document I'm using. Oh, let me big up Ziggy. That's tough. Ziggy, come back to me, try him out. Ziggy. Ziggy, let me try you out, Ziggy, man. Come on, man. It's even why I get the one in the Zigos. You know what I mean? Sunlight. But I don't know if you get the over thing right also. But you know this are the arrow. Chlorophyll. CH. Oh my. We can't even spell again, Ziggy.
Sir, CHOLROPH.
And yes. Chlorophyll. And then we say it is what, Ziggy? C6
Yes,
H.
sir. C6H12
Mm-hmm.
O6 and
Mm-hmm.
6O2.
Mm-hmm. All right. Big up Ziggy. Up top. Then we say state 4 ways in which a leaf is adapted for the process of photosynthesis.
Yeah.
One way from everybody. So watch out now. Espa, you're gonna start off, because Espa, you have no code name. You have a nickname with them call it. Thugs them call it. I want to have
Uh,
thugs. I sent
right.
Vincent.
So first one is equipped with
Mm-hmm.
stomata, so that's pore-like features on the underside of the leaf.
All right. So we have stomata. Presence of stomata. And what is the stomata again, Sasha? Active.
There are like those holes that in the leaf.
And what is the purpose of those holes in the leaves, Mama?
Um, transpiration.
Mm. More, more oxygen, more gaseous exchange, Sasha. So the stomata allows in the plant, it takes in carbon dioxide and let out CO2. Make sense?
Yes, sir.
So it is the holes, as you call it, that exchange allows efficient gaseous exchange. Carbon dioxide in, oxygen out. Make sense? Mm-hmm. So, Sasha, see you on the floor. We're still going with you. Give me
The
one way.
The leaves are thin,
Ah, nice, Sasha, nice. Thin leaves. And what does thin leaves allow for?
enable gaseous exchange.
And what's the main reason why it's thin, apart from gaseous exchange? Because really gaseous exchange why it's thin, you know. It's really thin because the gaseous exchange. Yeah, I mean, you're not wrong, you know, but more specifically, it's thin for another reason. Because it allows light to go to the chlorophyll easily. It allows the sunlight access the chlorophyll easy. Hmm? Make sense?
Yes, sir.
I feel bio-ish today, you know. Bio, my favorite subject, you know. So I
Yes,
can't feel it, you know. I love bio, you know. I love bio to art. Okay, for the fusion. All right, me see you, okay, okay. I see you. All right, then. We're going to a book now. We're going to a book. We may have two ways. Ziggy, the people them on YouTube want to hear from you, my general. The man them want to hear from you. You're the star.
Yes,
E-
sir. Yeah.
Every time I say Ziggy, the man them I say up top. So you hear me I say no. I hear a time.
Big up YouTube. Yeah, yes, sir. Sir Vince for support.
Being for support. That's like
Yes,
about
sir.
trying. But veins for support. Explain more.
So like for hold up the leaves to get sunlight, hold it up properly.
Mm. You know, say if you explain that, you know, like that, you know, you could have thief a mark, you know, Ziggy. You know, you
Yes,
could
sir.
have thief. You could have thief a mark still, you know. But all right, it makes sense. You know, textbook
Yeah.
like that still. But it makes sense. It makes sense.
Yes, sir.
Yeah, I mean, it makes sense. But you wouldn't want to say
Say
vein though. I
say your
mean,
reason.
yeah, you know, I mean, me see where you come from. But we want something more.
Yeah.
May I get something more safer? Yeah?
Yes, yes, sir.
May I get something more
Uh,
safer now? So I could give you large leaves, large surface area upon the leaf. The
mm,
leaf them
yes,
broad,
sir.
them big, large
Mm,
leaves. You know what I'm saying? So large
yes,
leaves,
sir.
make sure say what got them.
Say that the light gonna catch the leaf
At
properly.
you, at you, at you. So large surface area means that more sunlight for the plant, and more sunlight equal more food, more photosynthesis, active Ziggy.
Yes, sir.
One more, volunteer. Who want go? Come on, no, man. Not active, you know, man. Active, 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 active. I love the activity, you know. Man, I wanna pay on a nice little money, man. Active, let's go. Some people on YouTube wish them there, you know. They're not there. But them are going to watch and still without the interaction.
Yeah.
Yes, let could go.
The leaves contain chlorophyll to trap sunlight.
So the leaf contains chlorophyll to trap sunlight. But all right, now, I'm going to fix up that now, Sasha. I'm going to fix up that, you know. You ready for you fix? Watch out now. So which part of the leaf, which part of the plant make chlorophyll?
Chloroplast.
Chloroplast. So therefore, you'll say, Sasha, instead of say contain chlorophyll, let's say has a lot of chloroplast. Then, once it have chloroplast, it have chlorophyll. And they active. You know, Sash?
Yes, sir.
Active. So we see that. And then some people, we can also add waxy cuticle for water retention. We know about the waxy cuticle. Big up that answer from YouTube. Yes, waxy cuticle. We know that. We know what the waxy cuticle is, guys.
Yes, sir.
Right, so in a hot time, we know so the waxy cuticle, prevent water loss. Yeah? Yeah. But these answers, you get them answer you're active and you're active. Next. Yeah. We see another graph question. So this question says, a farmer who is growing his tomato crops in a greenhouse is concerned that the yield will be low supply. This is like a past paper question. I'm sure I see that question somewhere. But anyway, let's go. The question says a farmer who is growing his tomato crop in a greenhouse is concerned that the yield will be too low to supply the expected market demand. Right? The farmer therefore monitors the rate of photosynthesis of several plants, and the results are shown in figure 4 below. So we got some book. We see rate of photosynthesis, we see light intensity, we see a little bend so you understand? Then now we see explain the shape of the graph in figure 4. All right, so Espa, I don't want you to hold on for your answer, because Espa out the door from there, So since you're out the door from there, I want to hear from Sasha. So Sasha from Guyana.
Sir,
Big up Sasha
I
from Guyana. So Sasha from Guyana, people want to hear from you, Because me alone I teacher, Me are the facilitator. So go on now. Explain that to me now.
sir, as the light intensity increased, the rate of photosynthesis increased, then which the rate of photosynthesis remained constant due to the limiting factor, which is carbon dioxide.
All right, so what you know? So we are saying now, so the first part I say, the rate of photosynthesis increase as light intensity increase. That you say, right?
Yes, sir.
And then go on after that. No. I could follow you.
Then the rate of photosynthesis remained constant.
No, all right, so that's a no. That's a no. We need to ascertain the points at which that do so. And then remember, Sasha, reach at the leveling part yet. Remember the style we use in the other graph are the same style we are come back over here so with. So after we say that light intensity increase, right? We are go always in a graph like this, even though there's no degrees or there's no start, there's no specific timer or whatever variable there is, we are going to create one. So we are going to say at the beginning or at the start, right, the rate of photosynthesis increase rapidly with an increase in light intensity. Cool? But remember, Sasha, when you talk about the graph, and you say what you see, this is what you're seeing, Sasha. So why is this? Remember, you have to always say, why is this like this? So why is that like that? Why is the rate of photosynthesis increase rapidly with an increase in light intensity?
Because the leaves trap more sunlight, which increases the photosynthesis.
Yes, but yes, that is good. That is good. But you missed out something nice and sweet that you could say. Because there's absence in what? Why does the leaves take in more light? Because there's absence in what?
Sir, are you sure?
Because there's absence in there's no cloud. Is there a cloud?
No, sir.
Is there a breeze? Never said so. So what are these factors called? There's none of these clouds, there's none of these factors. So there's absence in what? Sasha, the people
Is
them
it
want, you know. The people them
humidity?
want, you know. Eh? There's an absence in limiting factors. That's all. You get what I say?
Hmm.
Remember, what are limiting factors? Once we know that? We know that, right? No?
So limiting factor.
Yes. Limiting factors are the factors that would decrease it. So if there's no sunlight, it would be absence of those factors. So if there is clouds, let's say there's cloud, there's a limiting factor. Right, Teresa?
Yes, sir.
Right?
Yes, sir.
So all of these are limiting factors. So there's no limiting factor here. Let me read that again. Light intensity is going up. No limiting factor. Right? So limiting factor is taking its time to be decreased. Because remember, more than like this is a morning thing. Right? Let's think of it as morning. When the sun just forward out, is the sun fully active?
No, sir.
When does the sun becomes fully active?
12 p.m. to 3
Yeah,
p.m.
man,
Roughly.
midday, right? Roughly midday. So that means that when it reach midday, there's going to be the as the time increases, it's going to be the hotter it becomes because the limiting factors would be taken away. That's all. So yes. So we will say at the start, the rate of photosynthesis increase rapidly with an increase in light intensity because there are less limiting factors. And therefore, there are more light, more light provides more energy for photosynthesis. Are we
So
there,
could
Sasha?
you relate,
Yes, go ahead, Espo.
so could you relate it to opening and closing of the stomata as well?
You could, depending on how you explain it. You could. Because it could be a limiting factor. So you could say the stomata, but you don't really have to say stomata, because there are many limiting factors. Once you say limiting factors, you cover every potential limiting factor there could be. You get what I'm saying?
Yes, sir.
And just bear in mind, guys, what you're saying is not wrong. Just bear in mind. However, with me, I'm a little bit of a perfectionist. So me I go always want you to attempt it, right? With the best you can. Then we reach a level enough now, Sasha. You're up again.
Is where we there?
We are in we are at the leveling off now when it becomes level. It start to be stabilized.
Oh, the limiting factor, carbon dioxide.
Okay. So go on now. You're saying the limiting factor carbon dioxide.
Causes the rate of photosynthesis to remain constant.
Yes. Despite the light conditions. Because remember that throughout the day, there would have been more light intensity would have been increased. Right? But because the limiting factor of carbon dioxide enters, right? And remember that temperature, remember that in this case, it is saying that the 33 degrees Celsius will be what? Will be what here? What will be 33 degrees Celsius will be here?
So I zone out and I forget what's going on.
Yes, the one out? Come back,
Yeah,
sir.
for a little minute.
Come back home, sir. So we are saying that at the start of this, the rate of photosynthesis increased rapidly with an increase in light intensity. This is because limiting factors decreases, I should say here, my bad. Somebody couldn't tell me say a degrees maybe so. Decreases and more light and provide more light. And more light provides more energy for photosynthesis, right? Then we are at the leveling off. So Sasha said that the rate of photosynthesis remain constant, right? Because limiting factors such as carbon dioxide concentration, right, has entered the frame. So I'm asking you now, the 33 degrees Celsius that's there is an example of what would be the 33 degrees Celsius there.
So it's also a limiting factor.
It's also a limiting factor. And it's also, you could argue that based on the graph, it is optimum. Based on this reaction, it is saying that anything below 33 or above 33 degrees, there would not be an increase in, there would not be an increase in the reaction. You understand? I will last.
I understand.
Sasha, we understand? I will last.
There something like that.
You last?
I'm not last, but I'm trying to understand. I'm getting
All
there.
right, let's go again. So, whoa. Okay. Okay, let's go again. All right. So let's start from the bottom. So we are saying here that right here we see where from the start the photosynthesis increase as light intensity increase. Are you with me so far?
Yes, sir.
And then we are saying now that this is so because the limiting factors are also decreasing. So with limiting factors being decreased, the rate of photosynthesis would increase. Are we there so far?
Yes, sir.
Okay. So then now, after we acknowledge this, right, we are going up to where it is being leveled off. And when this is being leveled off, we assume that this is where we have optimum temperature. This is the highest point any reaction can go. Because remember, you know, enzymes are responsible for all reaction. You have photosynthesis enzymes as well. Right? So we are saying when it reach optimum, right, it would remain constant. It would not increase. because at these levels at 33 degrees Celsius, the temperature, that is where we can assume that the highest level of photosynthesis can take. This is where this is optimal. It can't go beyond that. So even if it is 35, 36, 37, 38, 39 degrees Celsius, you would not see it increase based on this chart. You would just see it keep level. There's no increase despite the variable of temperature. You with me now?
Yes, sir.
Right. So we are saying now, after a certain point, the rate of photosynthesis remain constant, even with further increase in light intensity. So we can see here where we are assuming based on this graph that the light intensity is going this way. And the graph is also going this way. So that means that the further you go is the more light intensity you have. But we see where the reaction remains the same. True?
Yes, sir.
Okay. So then now that's what we're saying. So despite the light intensity increasing, right, we are seeing where the rate of photosynthesis does not increase. Seeing that? Seeing that, guys?
Yes,
Yes,
sir.
sir.
Okay. So we are saying that, I don't know why this is stuck in. Oh, okay. Sorry. So we are saying that the rate of photosynthesis remain constant even with further increase in light intensity. And why is this remain constant? Because now there is the factor of carbon dioxide concentration of the 0.3%. And the 0.3% of carbon dioxide or the temperature 33 degrees Celsius has become a limiting factor. So the plant has already photosynthesized at its maximum under the given rate condition. So it already reached maximum. It can't go no more than that. It's like trying to buck a radio. Even if you plug in the radio in at 20 different sources, it only can go at a certain level and it cannot pass it. Are we clear now?
Yes, sir.
Sasha? Ziggy?
Yes, sir.
Yes, sir.
So you see something like this, you can match it. Good. Next question says, indicate by drawing a new curve on figure 4, right? So indicate by drawing a new curve on figure 4 the change in the rate of photosynthesis expected if carbon dioxide is pumped into the greenhouse. So indicate by drawing a new curve on figure 4 the change in the rate of photosynthesis expected if carbon dioxide is pumped into the greenhouse. What would that be like?
Sir, it would
So
increase.
yeah.
It would be what?
Increase the rate of photosynthesis.
It would increase the rate of. Can you draw the curve for me?
Yes, sir.
Let me see if I'm seeing it. Okay. Okay. So you are saying if the light goes above, it will go to drops. All right.
Yes, sir.
All right. We agree with this curve. Espa, would you do something different? I agree with this curve.
Is about right. Yeah, there's a steep gradient and then it levels off a bit after. Is about right.
Yes, I do agree with that curve. Just that make sure you level it off a little bit more with the ruler, right? So it look more level.
Yes, sir.
I agree with that curve. All right. Next one says plants do not photosynthesize when left in a completely dark environment. So we look for dark and light reaction. In the greenhouse, the farmer noticed that the shady plants are taller and have smaller leaves than the plants located in sunny location. Explain why the plants left in the shade did not die immediately. Oh man, this is so soft. All right. Common sense question. Let's go. You too. Me look for an answer too. I'm looking there as well. Let's go, guys. Why do you think so? I remember, guys, what time now? 2:19. Yeah, man, we have to speed up this,
Sure.
so we have to interact more because, yours truly not eat yet. Yours truly have to get the food. On the IT session later, then yours truly have to go eat.
The leaves will be smaller because no photosynthesis in occur
So the leaves are smaller, so what?
because no photosynthesis in occur.
No photosynthesis. You sure you want to say no photosynthesis? Remember, when we're talking about reaction, we're talking about enzymes. Remember, I said enzymes denature, they slow down, but they don't stop.
Oh, yes, sir.
Right? Remember that.
Said, yeah,
All right,
sir,
so
said the rate of,
let's look at what the question you need to answer. You need to answer why the plants left in the shade did not die immediately. So first, the plants do not photosynthesize when left in dark room. They are telling you that plants don't photosynthesize. Yes, KK. KK, you're looking right. You're looking good so far. All right, so plants do not synthesize when left in a completely dark environment. In the greenhouse, right? The farmer noticed that the shady plants are taller and have smaller leaves than the plants located in sunny conditions. So explain why the plants left in the shade did not die. That's the main part you need to answer. Why the plants in the shade did not die.
So you can relate to tropisms, like phototropism.
Mm. Mm. Mm-mm.
uh,
Yeah,
sorry,
overthink.
So because it says the shaded plants are taller, so that's why I
That
assume.
they left it Troy Esper. This is a classic
sure,
misdirection question. Remember, no, Esper? Some part of the question are pure noise, distractions. Focus on this. Explain why plants left in the shade did not die. That's all you need to focus on.
Because you got some light, even if it's a little bit, if they're in the shade.
Because they got what?
They still got some amount of light, even if they were in the shade.
Mm. Mm-mm. Not quite.
Sir, it uses up its stored energy.
Yes, yes, Sasha.
Yeah, that was good.
Yes, yes.
yeah,
If
for
it
real,
was
for real.
if it was in the dark, right? In the dark, shady plants are taller and have some smaller leaves than the plants located in sunny conditions. Explain why the plants left in the shade did not die immediately. Right? So they did not say why the leaves are taller than the ones that are in. They did not ask for a comparison. They're just asking you for why the plants left in the shade did not die immediately. So all them tall and smaller leaves and them something, they are pure distractions. Right? So one of the reasons is because the plants left in the shade were able to survive using stored energy, right? That were produced when they were in the light. That make sense?
Yes, sir.
All right. And these can be stored in their root, in their stem, anywhere. What is another reason now why they did not die immediately? It's 2 marks. And you have to put so much things, of course. Say you put they
So,
did not die because they were stored energy. What else?
so I don't know. Like I use an educated guess, but before you were
Yes.
talking about limiting factors, so I
Mm-hmm.
guess while the plants in the sun may have access to light, there may be more limiting factors in their environment compared to the ones that were in the shade. Because if there's
Mm-hmm.
a deficiency of sun, that's a small limiting factor. They might have other conditions that are better than the ones in the sun.
Mm-hmm. And that is true. While that is true, that's not why they would live longer, though. That's not why they would die immediately, though. Think about light and dark reaction. What do plants do when they're in the dark?
Yeah,
Respire.
they try to grow towards light.
Respire. Who said that?
Me.
So that if you say my boss, and they say if you think they're asking you light and dark reaction. That's so. So respire. So they will continue to do respiration and then use the energy created from that. Reactive or inactive? We get that?
Yes, sir.
All right. All right. So we reach a section B. So unfortunately, we're gonna call it here for the YouTube. So YouTube, big up on yourself. I hope you learn something. Stay tuned for some videos and them thing, them more classes and thing over the weekend. You know it go. But if you're here, you want to join our classes, number in the description, number in the description below the link. So you know so you can run on. Alright. So big up on yourself, subscribe, share the page, share the love with people who probably need the help. And then we'll link when we link. Alright? Keep the notification